So the goal of this whole experiment is to get a smaller spot at the focal point. And so to do that, we set up this system that has a beam expander to make a larger beam waist at the output. And because the beam waist at the focal point is inversely related to the beam waist of the system, we wanted to increase the beam waist in general to have a smaller spot at the focal point. So we inserted a spatial filter into our system to clean up the Gaussian spot. So the incoming beam has a Gaussian pattern, and when it goes through the spatial filter, it cuts off the messy parts, and there's a Fourier transform relation to have a really nice, clean Gaussian output.
To start off this lab, we placed a microscope objective that has an NA of 0.25 and a focal length of 16 in this special holder, which can also hold a spatial filter and has X and Y adjust for the spatial filter and a Z adjust for the microscope objective. We align the microscope objective without the spatial filter in it to the laser using X on the stage and Y, making sure that it was aligned all the way down. Then we placed it 200 millimeters away from the laser. Then we placed a 10 micron pinhole or spatial filter in the device and adjusted it until we got the center. So you can see here we aren't seeing any rings, and we're getting a very clean spot.
So in adjusting the spatial filter, this is what we want to happen. We want the center of our spot to be right in the center of the pinhole. So it's aligned in 3 space, in Z, X, and Y. The way to get it to happen is we start far away so that we can see something through the pinhole. And then we maximize for brightness, adjusting X and Y. And then we bring it closer a little bit and readjust, maximizing for X and Y. And continue that process until we have the best spot.
Alright, so for the next part of our lab, we had a 250mm 2-inch achromat which we placed at a distance away from our spatial filter in order to try and get light collimated out of the lens. And so to see if it was collimated, we used a shear plate interferometer and tried to get the fringes on there to line up with the dark line you can see down the middle. You can see that it's pretty straight, but there's also some aberrations in here, which we thought might have been an artifact of either our spatial filter or our microscope objective. So we swapped both of them out a couple times, but we still kept getting these sort of aberrated fringes. No matter what we did and no matter how perfect the spot was coming out of our spatial filter. So that would seem to indicate something is wrong with our laser. A possible explanation for why our shear plate exhibited lots of aberrations when we looked at it was that we thought something was wrong with the laser, and it's possible that because these lasers are kind of old that one of the mirrors has become a little askew, which would induce aberrations.
So we're testing the sensitivity of the shear plate interferometers. By doing that, we're adjusting the micrometer that adjusts the spacing between the microscope and the spatial filter, and seeing the range at which the lines on the spatial filter are still parallel, so it's collimated. And we found that the larger thing is 20% more sensitive than the smaller one.
We wanted to measure the percentage of the original power that made it through our beam expander, but unfortunately our beam is quite large, so it was much larger than our detector size. We placed a second lens behind the expander, and then from here we shrunk down the beam so that it fit onto the detector. Then we measured behind this lens, and we also measured it before the beam expander, and we found that we had 94.2% of total power traveling through our beam expander. By placing this lens in the system, we are losing a tiny bit more power than we would be without it, but that's a compromise we had to make to shrink the beam down to fit on the detector.
So for the last part of the lab, we used the same setup as in the previous lab with the knife edge. We have a photodetector and our multimeter. We used a piece of paper here to block off the middle of the knife edge. There's actually a hole in it, so you could get light refracting or diffracting off of that back in the detector. And then we covered it with this again. And then we took readings all the way through using 100 micron increments and plotted it in Excel. So we wanted to make sure the razor blade was perfectly vertical so that when you're moving it, translating in this direction, it actually is moving across the spot in the way we wanted it to. And then we took 2 readings, one before the beam waist and one after the beam waist.